On April 4th, 2017, a privileged group of telescopes on mountains across the planet switched on at the same time. For the next week, they danced in unison, collecting radio waves dispatched from the center of our Milky Way galaxy and from the galaxy M87. Together, they make up the Event Horizon Telescope, a global project to capture the first ever picture of a black hole. That's right, ever since physicists first conceived of black holes centuries ago. Every image of one from our textbooks and our space agencies, they're all illustrations. Until now.
We are delighted to be able to report to you today that we have seen what we thought was unseeable.
For centuries, physicists have theorized that an object with enough mass and density could trap even light in its gravitational field. Just as you have to travel faster to leave Earth than you do to leave the moon, there could be a place where you'd have to travel faster than the speed of light to escape, and nothing moves faster than light. The math from Einstein's theory of general relativity describes an area completely invisible to us within a boundary called the event horizon. And at the center of that black hole is a singularity, a point of infinite density, which is where physics as we know it breaks down.
They showed up in the math long ago and they kept reappearing and they sort of persistently would not go away. But Einstein always thought that there must be some physical mechanism that prevents stars from collapsing to an infinitely small point, which is actually pretty reasonable. I mean, because it sounds insane.
Eventually, scientists began to see things that only made sense if black holes were real, like the orbits of these stars around the center of the Milky Way galaxy.
You see these stars just slingshotting around an invisible point. And a black hole is the most likely explanation for putting that amount of mass in that small space for something that's completely dark.
We can also see the glowing material that spirals around black holes. Friction heats this matter up tens of millions of degrees, and anything that hot emits X-rays that we can detect with telescopes that orbit above Earth's atmosphere. This is a pair of galaxies that pass through each other. There are at least 9 suspected black holes here, but you can only see them when you look at the X-ray layer. These dots are X-ray sources linked to suspected supermassive black holes at the center of galaxies 3 to 10 billion light-years away, and that's just from this small patch of sky. Some supermassive black holes also feature gigantic jets of particles, seen here in radio wave data from the galaxy M87, which has a much bigger black hole than the one in the center of the Milky Way. No other known source of energy could power these things, and nothing we know of besides 2 black holes colliding could have produced the gravitational waves we detected in 2015. Scientists think there are black holes, large and small, all over the universe. We can see their fingerprints, but we didn't have the mugshot. Directly imaging a black hole has been impossible because they're either too small, too far away, or both. Sagittarius A star, the black hole at the center of our galaxy, has the mass of 4 million suns, but it would fit inside the orbit of Mercury. Imaging it from Earth is like taking a picture of a DVD on the surface of the moon, with huge clouds of dust and gas in between.
So many things had to go right for this image to exist. So the first thing that has to happen is there has to be some slice of light that travels all the way from the edge of the black hole without getting knocked off course or absorbed by any of the gas or anything in between. And then it also has to make it through the Earth's atmosphere, which a lot of frequencies of light don't.
They landed on a wavelength of 1.3 millimeters at the high-frequency end of the radio spectrum. With that wavelength and with 8 observatories across the world, the Event Horizon Telescope had a chance at seeing a black hole, as long as the weather cooperated.
You have to have clear weather in all of those places at a time when the Earth is oriented in such a way that all of those telescopes can see the black hole simultaneously. They can really only observe once a year.
There was so much data involved that it had to be flown on airplanes. They waited 6 months for the hard drives to arrive from the South Pole, which closes during wintertime. This multi-telescope method is called Very Long Baseline Interferometry. It correlates time-stamped data from distant telescopes to boost the signal and quiet the noise. Each pairing of telescopes contributes a piece of the puzzle. But the image doesn't just pop out after that. They had 4 groups working for months to generate the image that best represents the data.
Each group was working individually and in isolation from the other groups, working with the same data to see that each group came up with the same image or not.
Here it is.
And the result of all that work is this. The bright parts are the matter and light swirling around the black hole, and it's brighter on the side that's moving toward us. And the dark part is the black hole's shadow, which includes the event horizon plus a region where light could escape but doesn't. The size and shape of the shadow appear to confirm the theory of general relativity.
Today, general relativity has passed another crucial test, this one spanning from horizons to the stars.
Humanity's first image of a black hole isn't crisp and beautiful like the illustrations or the movie Interstellar. It's better.
The picture we see this week is made of scraps and bits of light that's been traveling across the universe and collected by these, aluminum dishes on top of mountaintops and then combined in a supercomputer to make this image. So that's why it's real.
astronomers have begun one of the most far-reaching efforts ever undertaken to study the universe. They are forging giant new lenses and mirrors while marshalling vast computational power. These new technologies are at the center of a historic quest to peer into the deepest recesses of time, to find out how the universe set the stage for galaxies and worlds like ours. In an era known as the Cosmic Dawn. Philippe Menenteau and colleagues from the University of Illinois are part of a global push to advance the science of cosmology, the study of the universe as a whole.
Hey, hi guys.
They are mapping the positions of galaxies across the sky and extending deep into the universe.
Oh yeah, that doesn't look good.
Yeah, no.
So that was only at the end.
Their goal,
show us something
to link the evolution of planets, stars, and galaxies, the universe we see around us, with conditions that existed at the dawn of time.
That is because it's in a corner. Those are corners.
What forces came together to form the very first generation of stars and galaxies, and over time, the vast architecture of matter and light we see in our telescopes. Only a century ago, astronomers debated whether the universe is confined to a giant rotating disk of stars, dust, and gas, the Milky Way, or whether our galaxy is one of many so-called island universes. We now know that our galaxy is part of a formation of 3 major galaxies, along with some 51 dwarf galaxies called the local group. The local group is bound by gravity to a much larger formation, the Virgo cluster, with up to 2,000 galaxies. Beyond Virgo, galaxy clusters are linked to superclusters in a pattern laid out by the first great cosmic mapping project, the Sloan Digital Sky Survey, beginning in the year 2000. As the Sloan data shows, superclusters are connected to each other by streams or filaments of galaxies, bounded by immense empty regions. To understand how the universe got this way, astronomers must identify its basic components of matter and energy. Back in the 1930s, the astronomer Fritz Zwicky measured the rotation rate of spiral galaxies like the Milky Way. He found that the gravity that binds their stars is over 100 times greater than what he expected, based on the amount of matter that's visible. Zwicky called the unseen substance exerting this extra gravitational pull dark matter. You can see its influence on even larger scales. Without dark matter, the gravity of clusters like this is not enough to hold all the galaxies rotating around it. In this case, dark matter acts as a lens, magnifying and distorting the light of galaxies in the deep background and causing them to appear as blue arcs. Astronomers use the pattern of gravitational lensing to map the distribution of dark matter in the supercluster Abell 1689, shown here as a blue haze. On a large scale, galaxy structures like these are a measure of how much dark matter there is.
One of the tools that we have in cosmology to pin down the kind of universe that we live in is the growth of the structures. These are giant systems with hundreds to thousands of galaxies. And these systems, these ones that are particularly big, are very, very rare. And the number of those that you can find as a function of cosmic time, from here to the past, depends. It's a very strong prediction of the universe that we live on.
This computer model shows the role dark matter has played in shaping the contours of the universe. Not long after the Big Bang, gravity began to amplify slight initial variations in the distribution of dark matter. Regions of the highest density attracted enough visible matter to form galaxy clusters and the largest superclusters. Even as astronomers struggled to define what dark matter is, they discovered another mysterious and powerful influence on cosmic evolution. In the late 1990s, two groups of astronomers asked whether there's enough dark matter out there to one day slow or even halt the expansion of the universe. Using the Hubble Space Telescope, along with ground-based telescopes, the team set out to track the rate of cosmic expansion through the entire history of the universe. They did this by searching deep space for a particular type of exploding star. Called a Type 1a supernova, it often begins with two stars in a close orbit, one of which draws gas from its companion. When it gains enough mass, it undergoes a runaway nuclear reaction and explodes. Because Type 1a supernovae are all thought to blow up in the same way, they have the same intrinsic brightness. That makes them ideal for measuring cosmic distances. It's like looking at car headlights approaching on a highway. The dimmer they appear, the farther away they are. The astronomers combined distance measurements with another marker, how far their light had shifted toward the red part of the light spectrum. The greater this redshift, the more the universe had expanded since the star exploded. Some explosions appeared dimmer than the teams expected based on their redshift. That meant their light had traveled farther than it should have, given a constant rate of expansion. This finding led the two teams to conclude that, in the deep past, the universe was slowing down. But to reach its current size, it must have then sped up. Scientists now believe the universe is dominated not by matter we can see, nor by the mysterious gravitational presence, dark matter. It's something else, pervasive and powerful enough to cause space to accelerate outward. They call it dark energy. One leading idea is that it stems from particles that well up from the vacuum of space. As the universe expands, it generates more and more dark energy. That has the effect, over large distances, of counteracting gravity and causing space to expand even faster. Spread over the vastness of the universe, it is the energy equivalent of only 5 hydrogen atoms per cubic meter. And yet scientists find that dark energy accounts for 68% of the entire cosmic matter energy budget, with dark matter at 27% and ordinary visible matter less than 5%. The twin discoveries of dark matter and dark energy have thrown cosmology into turmoil and potentially into a new age of discovery. Astronomers have now launched a full-scale effort to pin down the forces and events that drove cosmic evolution, going back to the earliest times. Already, a fleet of space telescopes led by Hubble is scanning the distant universe for light sources across the electromagnetic spectrum. The long-awaited James Webb Space Telescope, slated for launch in 2018, represents the next generation of great space observatories. With a segmented primary mirror that is almost 3 times larger than that of Hubble, the James Webb will capture the trickle of photons from a time nearly 13 billion years ago, when the universe lit up with stars and galaxies. The next generation of ground telescopes will radically extend our light-gathering power. The European Extremely Large Telescope, now under construction in Chile's Atacama Desert, will have a 39-meter mirror that quadruples the light-gathering power of the largest telescopes today. While these telescopes extend our vision into the deep universe, the American W1 and European Euclid space telescopes will take in large regions of the sky at high resolution. These new instruments, each slated for launch in the 2020s, will be used to survey the deep universe for Type 1a supernovae and other markers of cosmic evolution. These space observatories will work in conjunction with an ambitious new effort to track the evolution of the cosmos in near real time. The Large Synoptic Survey Telescope, or LSST, is being built in the mountains of Chile. It combines data gathering on an unprecedented scale with plans for dedicated fiber optic connections capable of delivering a flood of data to supercomputers a continent away. From there, it will be processed and made available through advanced internet links to scientists around the world. At its heart, the LSST will be outfitted with the largest digital camera ever built.
I don't see any sleeps.
That includes the largest lens ever built, now undergoing final polish before assembly.
Good, huh? Yeah.
With a field of view the size of 50 full moons, the telescope will observe just over half the sky visible from the Earth, to a depth of about halfway back to the beginning of time.
LSST is going to take a new image of the sky in roughly every 47 seconds. And so For us to bring the picture of the sky to the world as quickly as possible, we are going to release a catalog of how the universe appears to have changed with time within 1 minute of that shutter closing. And so every 47 seconds, every night for 10 years, we're going to distribute effectively a mini worldwide data release of how the sky looks like it changed with time. And on average, LSST will image the entire southern sky roughly once every several days.
Astronomers expect the telescope to record a blizzard of transient events, from asteroids buzzing through the solar system to black holes flaring up in distant galaxies and stars exploding out on the horizons of space and time. The telescope will revolutionize what scientists call time domain astronomy.
Right now, there are lots of time domain surveys going on, and worldwide you might get thousands of new alerts per night of things that have changed in the sky. But LSST will change that. It is so much bigger and samples such a larger volume of space that it will have 10 million new things every night that it looks at the sky. That's 10 million every night for 10 years. And so suddenly it's a whole different ballgame, right? The scale is well beyond anything that has ever happened in time domain astronomy. It opens up space that we've never explored before.
To make sense of the deluge of data, this project will make extensive use of supercomputer models designed to simulate periods in cosmic history. These powerful programs are based on theories of star and galaxy formation, the influence of dark matter and dark energy, and a host of other parameters. Scientists will use them to test theories about what drives cosmic evolution by comparing simulation results with data captured by the telescope.
These data sets are so complex that we really need simulations of cosmic evolution to even understand and analyze the data, let alone interpret them. The computations really translate our theories of dark energy, dark matter, cosmic evolution into observables that we can then go out and test with our observations.
The LSST project will build upon previous large-scale cosmic surveys. The Sloan Digital Sky Survey has mapped galaxies out to 1/3 the distance to our visible horizon. Astronomers are now leaping beyond that with a project based on a summit across the canyon from LSST. It's called the Dark Energy Survey. Here, at a dedicated 4-meter telescope, Felipe Menento and colleagues are pioneering new systems and procedures for mining the light of deep space.
515-473.
515-473?
Yeah.
Because of the time it takes for the light of distant objects to reach us, when these astronomers look deep into the cosmos, they are looking back in time.
We are looking back in time, kind of like an archaeologist, like digging deeper into the ground. And at each one of these epochs, we are kind of like seeing the relic, the fossils that were left. So we cannot follow a galaxy back in time, but we can actually take snapshots of populations at different cosmic times and see how they have been changing since early on until today.
Yeah, but the thing is that, under this, there are...
2012B.
No, you're not that. You're like 2016A.
The telescope captures the light of stars and galaxies across the electromagnetic spectrum, from high-energy ultraviolet to low-energy infrared. These colors reveal important galaxy characteristics, such as the rate of star birth, the amount of dust or gas within them, their distance from Earth. The camera's sensor divides the field of view into 62 high-resolution detectors. Each one captures countless thousands of celestial objects, some bright and well-known, others too subtle to see with your eye. Night after night, month after month, the exposures pile up across a survey area that covers 1/4 of the southern sky, or 1/8 of the entire sky as seen from Earth. With data from the Dark Energy Survey combined with a much larger LSST survey, scientists will create a three-dimensional map of galaxies going back to when the universe was half its current age.
We only understood that we live in a universe full of galaxies in 1930s. Before that, we didn't understand our place in the universe. And since then, there's been this constant quest to understand why galaxies look the way they look. How did they form? And how this process has been shaping also, the planets, the stars, everything that it's in them. Because, galaxies are the building blocks of the universe.
In recent years, advanced telescopes have shown that the universe is filled with galaxies in a wide variety of shapes and sizes. From giant spheres of ancient dying stars to complex, twisted shapes, often run through with rings of dust. The historic Hubble Deep Field took us back for the first time to the early stages of galaxy formation. It found that blurry scraps of stars and gas visible at the dark margins of space are primitive galaxies. Theory says they will one day merge into larger, mature galaxies.
Something that astronomy has been trying to answer for decades, particularly after the Hubble Space Telescope was in space and we were able to see with amazing precision the morphology and the shapes of the earliest galaxies in the universe. We've been trying to answer and trying to connect morphology and colors with the evolutionary stage of the galaxies.
Okay, you have this kind of galaxy and that kind of galaxy. How do they fit together? Or do they fit together? Does that galaxy turn into this galaxy or the other way around? Does this kind of galaxy never become that kind of galaxy because it didn't have the right nurturing or the right environment? If you were an alien coming down with no knowledge of how humans work, and you landed in a city, and you were just walking around looking at some city blocks, you would see all kinds of different people. You'd see babies, and you'd see old people, and you'd see teenagers and people in their mid-20s, but you wouldn't necessarily have an idea of how all those people fit together. Do people just arrive at these different stages, or are they working through some evolutionary process? So it's the science that lets you understand that, things are evolving. So you have a baby that then grows to a toddler, that grows to a teenager, that, eventually becomes an old person.
Using the ALMA telescope array in Chile, astronomers caught a glimpse of galaxy evolution in its earliest stages. They focused the telescope on the southern constellation of Cetus, setting their sights on a seemingly empty region. Deep within it, about 3.5 billion light-years from Earth, lies the galaxy cluster Abell 2744. It's known as Pandora's cluster for the tangle of shapes created when at least 4 smaller galaxy clusters merge together. To one side, astronomers found a faint, ghostly shape that had been magnified by dark matter within the cluster. It is a pocket of stars, far beyond and much older than the galaxy cluster. The stars were being born when the universe was just 600 million years old. This animation recreates the ancient star cluster, surrounded by gas and punctuated with supernovae. Over time, most star clusters like this would have merged with a galaxy. As it turns out, a small number have managed to stay intact over the billions of years since they were born. Finding them within their original dark matter cocoons has become a passion for these members of the Dark Energy Survey, Alex Derlika-Wagner and Keith Bechtel.
So we don't forget.
There are expectations from this model of galaxy formation for the existence of many of these small dark matter clumps in the halo of the Milky Way. And so while this was an expectation that was put forth, basically from simulations, there were very firm predictions about if this paradigm were correct, how many dwarf galaxies DES should find.
Astronomers have long known that the Milky Way galaxy is enveloped in a diffuse halo of stars, including some 160 large star clusters. M15 is one of the densest known. Gravitational interactions among its stars have caused them to pack in tightly. So-called globular clusters like this are like pottery shards found by archaeologists at the sites of ancient villages. One, called Turzan 5, has even managed to survive a fall into our Milky Way. It contains a population of relatively metal-poor stars that would have been born 12 billion years ago. There should be many more clusters like these, in a wide variety of sizes, that have simply not had time to enter the disk. Where are they today? When the first round of data from the Dark Energy Survey was released, Alex and Keith began combing it for light that could be resolved as stars. They saw what they were looking for. Tiny remnants of the Milky Way's birth, star clusters almost entirely devoid of metal content. These dwarf galaxies turned out to be dark matter rich, with about 10 times the ratio of dark to visible matter as seen in the galaxy as a whole.
We have this idea that galaxies form from the bottom up. Many, many small galaxies, and then, over billions of years, they merge together to form larger galaxies. What this means is that the smallest galaxies were also the first galaxies, and therefore they're the oldest. And when you actually look at these dwarf galaxies and you study the properties of their stars, you find that the stars are very, very old, that most of them formed over 10 billion years ago.
This realization is central to cosmology's quest to link the early stages and growth of galaxies to cosmic evolution on the largest scales. That quest points to the ingredients of matter and energy that produced the very first stars at the cosmic dawn.
Immediately after the Big Bang, the universe was hot and very, very, very homogeneous, but it wasn't perfectly homogeneous.
In fact, astronomers have found a telltale pattern in light emitted when the universe was just 300,000 years old, the so-called cosmic microwave background. In this image from the European Planck satellite, the colors indicate hot and cold patches produced by tiny variations in the energy of the Big Bang.
These small fluctuations, these small variations of matter then got amplified by gravity. And these tiny variations, this tiny clumping of matter were the seeds of the galaxies that we see today. And this is crucial. The amount of variation that we saw early in the universe predicts, together with all of the other ingredients that we need for a universe, predict the rate in which the structures are growing. Meaning the number of clusters of galaxies, the number of superclusters, the shapes of the cosmic wave is determined by that initial imprint.
To trace the evolution of this imprint, scientists are using a supercomputer model to recreate the eruption of stars and galaxies in the cosmic dawn. It begins in the darkness of the early universe, barely 6 million years after the Big Bang. Gravity draws dark matter into diffuse halos. Within them, hydrogen gas forms clouds that become more and more dense over time. As gravity compresses the clouds, they begin to heat up, then finally ignite to form the first generation of stars. These stars are giants, much larger than any today. One blows up in a powerful supernova. The model shows an environment transformed by the explosion. The supernova litters its surroundings with heavier elements created in nuclear fusion. Carbon, silicon, iron, and more. These so-called metals cause surrounding clouds of hydrogen to cool. That allows them to collapse. Turbulence breaks them into smaller pockets. A cluster of smaller second-generation stars now begins to form. Here's a wider view of the scene almost 400 million years later. From data generated by the simulation, scientists are working to isolate the dynamics of galaxy evolution. Stars are being born where filaments of gas, shown in blue, come together. Ultraviolet light from these stars begins to strip electrons from hydrogen atoms in a process called ionization. That causes surrounding regions to glow with visible light. The ionized gas appears as bubbles. They are associated with pockets of elevated temperatures, shown in red, as well as high concentrations of metals spread by supernovae, shown in green. The simulation reveals a dynamic that shaped the course of cosmic history. Heating from ionization tends to push the gas out. That suppresses the rate of star birth. Metals, on the other hand, allow pockets of gas to cool and fall inward. That increases the rate of star birth. So instead of stars forming and collapsing immediately into galaxies, the universe becomes a wide mix of hot and cold regions, large and small star clusters, and pockets of gas amid clouds of dust rich in metals. The small dwarf galaxies that astronomers have spotted hovering above the Milky Way are relics of this early period and of the galaxy's early years.
The formation of the first generation of stars, also referred to as population 3 stars, were these very massive stars to form early in the universe, polluting the intergalactic medium and the interstellar medium with unique chemical fingerprints of their own formation, different than the sorts of supernovae that we see today. And ultra-faint dwarf galaxies, we have evidence that many of them are actually fossils of this era of reionization, where some of them are thought to have formed before reionization took place. And we also have evidence from the chemical abundances of stars in the ultra-faint dwarf galaxies that the chemicals that they were enriched with may have been coming from that first generation of stars itself.
The supercomputer model gives us a view of cosmic evolution advancing to an age of about 1 billion years. The scene is dominated by star birth and by star clusters merging together into larger formations. The universe continues to put the brakes on galaxy growth. While star birth spreads heat, stifling the flow of gas into galaxies, metals from stars and supernovae have a cooling effect that enables this flow. Many of these early generation galaxies join in larger aggregations. Take the Spiderweb galaxy, 10.6 billion light-years away. A close examination shows that it sits in the middle of a cluster of galaxy fragments. This animated reconstruction shows the chaotic scene. Hundreds of small galaxies and patches of stars are interacting while drawing in matter from the surrounding region. Starting in the early years of the cosmic dawn, this simulation shows a slice of the universe in a region 350 million light-years across. The gravity of dark matter gradually concentrated visible matter into galaxy clusters. At the centers of large galaxies, black holes grew to supermassive proportions. As matter flowed in, they generated immense expanding bubbles of gas. These bubbles pushed beyond their galaxies, spreading waves of hot gas. The heating from these bubbles would slow the flow of gas into the clusters. That allowed smaller galaxies like ours to form on the margins. At the same time, black hole winds seeded the wider universe with dust and metals generated by supernovae. Flash forward to the present era. Our galaxy has by no means completed its evolution. This simulation recreates the last 60 million years of its history. Within the disk, each flash of light is a supernova. As time goes by, thousands upon thousands of these explosions seed the galaxy with metals, the cosmic dust from which new generations of stars and solar systems are born. Though most of the Milky Way's stars reside within the disk, some orbit far above or below it in the galaxy's halo and occasionally pass through the disk. Our galaxy today is the product of countless small and large mergers going all the way back to the early universe. Its landscapes are the ever-evolving product of star birth and star death. The Milky Way is filled with some 200 billion stars born at each stage in the life of the cosmos. They are intermixed with clouds of dust and gas, all swirling around a bright central region called the bulge. We glimpse its origins within a halo of stars and small clusters, some nearly as old as the universe. From our vantage on Earth, the universe continues to reinvent itself.
Okay. Así que vamos a hacer star floods. Okay.
A supernova's light has just reached Earth from a nearby galaxy called Centaurus A.
Not right now, it's taking an exposure.
Okay,
so
yeah, let's finish that one.
It's of particular interest to the astronomers. Its interaction with surrounding dust clouds can reveal the environment in which its parent star lived and died.
If you're a physicist, you have a lab, and in your lab you can change the parameters of your experiment and keep testing it. When you are an astronomer, you cannot create stars, you cannot create galaxies. The universe is your lab, and you are a humble collector of light.
Try 515473.
515473?
Yeah.
Okay.
Day by day, month by month, the light of the universe rolls into the data pipeline. Here is one slice of the southern sky from the Dark Energy Survey, extending roughly half the distance to the edge of our visible horizon. It's just the beginning of a grand cosmic census that includes galaxy clusters, galaxy types, rates of star birth, chemical abundances, distances from Earth, and more. When the data from this and the Large Synoptic Survey are combined and laid out in time, they promise a record of how the universe evolved since its early moments.
It is just jaw-dropping to me that humans can even undertake these big questions and figure out where we are in the universe. And as we see the universe changing with time, over the last 13.7 billion years, it gives us a sense of the cosmic structure formation events that have ultimately led to systems like the sun being formed.
Discovering the shapes and contours of the universe is only the first step in understanding how it came to be. Astronomers will sift the data for clues to the initial conditions that came together in the cosmic dawn. They'll test theories about the identity of dark matter and dark energy.
But there is also even this more fundamental question, which is, do these things even exist? I think the evidence for dark matter is quite strong. We see it really explains a number of different phenomena. Dark energy, I think, is our best current hypothesis for what It's causing the universe to speed up. But it's still on, I would say, shaky ground. Is dark energy just the energy of empty space? Or is it the energy associated with some new fundamental particle in the universe?
Assuming current observations hold up, astronomers in the distant future may produce a very different cosmic map, one that reflects a universe pushed further apart by dark energy. Many of the galaxies we see today will have receded beyond our horizons, becoming invisible from Earth. Our own Milky Way will remain intact, still enveloped in the dark matter that spawned it. Its halo will become increasingly entwined with that of the Andromeda Galaxy, our larger neighbor. It is now moving toward us at about 400,000 kilometers per hour. When the two meet, several billion years from now, their interaction will dominate our night skies. From a point of view unique to their time, those future astronomers will look out at the horizon and ask, how did it all come to be? Where does it end? We ask the same questions today based on our point of view at this moment in cosmic history. Our technologies are allowing us to see nearly to the beginning of time and to track the behavior of the universe on the largest of scales. And yet, the more we see, the deeper the mysteries become.
Evolution, the natural process that shaped all life and can lead from this to this. It's driven by random genetic mutation and natural selection. Random mutations create variation. Sometimes a change gives an advantage, like cells that can detect light to help seek out safer environments. Survivors pass along the changes to future generations. Further changes can occur over many generations and sometimes hundreds of millions of years. Fast forward to today. Now we have molecular tools that can allow us to alter the direction that life takes, including our own. The most accurate and effective tool yet for gene editing is called CRISPR. We can use it to essentially rewrite any gene however we choose, potentially fixing mutations that cause disease. The most common analogy for how CRISPR works is the find and replace function of a word processor. And with CRISPR, what used to take nature millions of years can now be done in a lab in just days. It is a groundbreaking tool with unprecedented power. It shows promise for curing cancers, ALS, and many other diseases. We've already engineered malaria-proof mosquitoes and may create mice immune to Lyme disease. But possessing the power to forever alter the future of life raises difficult questions. What could justify genetically modifying the ecology around us? Should we move beyond curing diseases to making other genetic enhancements? Are there any lines we shouldn't cross? How can we avoid the misuse of this powerful technology? And can humanity even agree on the future of our very own genetic destiny? Tonight we're going to hear from a range of experts working on different aspects of CRISPR, which stands for cultured, regularly interspaced, short, palindromic repeats. Wow, but we are not going to let that scare us, are we? No. So allow me to introduce our distinguished panel. Josephine Johnston is a bioethicist and lawyer at the Hastings Center. She works on the ethics of emerging biotechnologies with a focus on their use in reproduction, psychiatry, genetics, and neuroscience. So please welcome Josephine Johnston. He's a member of the U.S. National Academy of Sciences and has served on several national research committees studying the environmental and health effects of the commercialization of genetically engineered crops. Please say hello to Fred Gould. Samuel Sternberg is an assistant professor in the Department of Biochemistry and Molecular Biophysics at Columbia University. He is the co-author with Jennifer Doudna of A Crack in Creation, The Unthinkable Power to Control Evolution. Sam Sternberg. Neville Sanjana is on the faculty of the New York Genome Center, an assistant professor in the departments of biology and neuroscience and physiology at New York University. Neville Sanjana. So first we're going to start with an overview of the great potential of this technology, CRISPR, in terms of human diseases. Research is underway on a number of conditions, and I'm just going to throw it first off to Neville right away. Don't be surprised.
Yeah,
just
great.
tell us what the range of diseases is that CRISPR has the potential to cure.
Yeah, so I think in the last few years it's had just a huge impact across biomedical science, and some of the diseases are very common diseases. A lot of us know folks who have cancer, cancers like skin cancer, lung cancer, blood cancers. There's also a lot of non-cancer applications, I think with CRISPR-2, blood diseases like sickle cell anemia or beta thalassemia. And other diseases that are being worked on right now with CRISPR are some eye diseases, some diseases of the retina that result in blindness.
So I know that there's been a lot of research on mice and monkeys, but none of this has applied to humans yet, has it?
There are clinical trials either underway, depending on, in some countries already underway, and definitely in advanced planning stages in US and Europe and China.
I heard that China actually did use CRISPR for an eye disease, right? On one person. Do we know what the outcome was?
I don't think that's been published yet, but there's actually a number of clinical trials for cancer that have started in China where you use CRISPR to edit immune cells that actually get delivered back into patients to hunt down cancerous cells of the body. So I don't think it's too early to say how effective these treatments are going to be, but there's a lot of hope, I think, across the scientific community that there's kind of this new age of precision medicine using gene editing to treat some of these more common genetic diseases or cancers in a fundamentally new way.
Wow. So Fred, just as an overview, just to start the whole discussion, tell us about CRISPR's potential in plants and animals.
Yeah, well, especially in plants, there's the possibility of using CRISPR to change the genetic composition of some of our crop plants to make them more resistant to plant diseases. And that could decrease the use of insecticides or herbicides and also of fungicides and so on. So that's been a very important possible contribution. And with animals, the hornless cow that was recently produced. You know, the idea that...
The what?
Yeah. So, well, bulls will have horns, and typically you have to do sort of a surgical operation to get rid of the horns. So the idea that you could engineer these cattle not to have horns is something that has been done by regular breeding, but could also be done using those things. But a lot of these things are just starting to be considered and are not actually on the market yet.
You know, I did a story on 60 Minutes about 10 years ago on a program to bring back extinct animals. And I've read that CRISPR may be used in that kind of an effort. Sam?
I think there's a book that's coming out or has come out using CRISPR to resurrect woolly mammoths. Now, we had a discussion earlier, like how realistic is that actually? I would still put it personally in the box of science fiction, but the point is we actually, scientists have decoded the entire genetic sequence of the woolly mammoth from fossil samples, from preserved specimens, and you can compare the Asian elephant genome and the woolly mammoth genome, pinpoint all of the precise DNA changes between the two, and now use this powerful gene editing technology to actually go into Asian elephant cells and start changing individual genes one at a time from the version that modern day elephants have to the version that woolly mammoths once had. And so
then the
idea
elephant would give birth, right?
Well, that's the part that I think no one's worked out IVF in an elephant, and the gestation time of elephants is like 2 years, and that's the big question. Could you ever actually give birth to something that even resembled a woolly mammoth? I'd say not in my lifetime, I would guess. But if you read the news stories about it, people say 5-10 years.
Josephine, just as an overview, what are some of the negative aspects of this as a bioethicist?
Well, the first thing I would say is I think because it's a platform technology with so many different uses, it's not sort of just a good or a bad technology, and the uses really vary. But people who are speaking about some of the concerns that they might have across these different potential uses are concerned about a real range of things, from safety type questions about like how would we know, could people create sort of weapons out of using these technologies. That's one sort of really sort of very clearly dangerous and potentially frightening scenarios, and that people are looking at all the way across to questions around how appropriate it is for humans to have such a lot of control over the genes of our ourselves, our children, or animals, and to what ends, like what are we pursuing? What's the purpose of it? So there are a lot of questions raised right across those different uses.
And as we move forward, we'll be discussing the pros and cons. So we set the stage, but let's go back in history a little bit and talk about all the different ways that we were manipulating evolution and our genes before CRISPR, and for good or bad. So Fred, start out with us on the plants and the manipulation of plant genetics.
Right. So I think that many of you probably do know that your plants didn't just arise from nowhere. They were almost all bred by farmers and by technicians over thousands of years. And so we often use the example of teosinte, which is sort of a weed that grows in Mexico with a very small seed pod, and that has been over time changed by farmers who have just selected the best plants that had the most nutrition over time to bigger and bigger corn. And today we have modern corn as a result of that. So we also have the same thing with our animals, both in terms of farm animals, but also think about your dogs and cats. Think about the odd goldfish you can buy in a store. We've manipulated animals and plants for a long period of time.
And what about food? Who wants to tackle food?
Well, I think you just
Okay, Fred, come back to us.
I'll come
Tell
back
us
to
about
food.
what we're
Sure,
eating.
sure.
I mean, is it all genetically modified today?
Yeah, so I think it's a very, genetically modified, okay? So I just told you about teosinte being bred to become the modern day corn plant. That is genetically modified from teosinte. But when people talk typically about genetically modified or GMOs, they're talking about something that has been engineered. And we have to talk about the difference between that. Right? So, and I think a lot of it goes back to a cultural thing about what is natural and what is not natural. And it's natural to just save seeds that are better and better and move ahead. Most people will say that is natural, but they wouldn't say that it's natural to take cells out of a plant and insert DNA in them to make them different. And so we have about 80 to 90% of our corn, soybean, and cotton in the United States has been developed that way.
Genetically modified.
Genetically
GMO.
engineered. And I like to use the term genetically engineered because it's engineering, right, in the sense that it's actually thinking about an architectural plan of a plant and changing it, as opposed to something that you just do tinkering with it, as some people would say. So that's a big change in terms of how people relate to something that's been developed those different ways. And to go back and say that what's the difference between a plant that has the same characteristics? Let's say you change a corn plant from being yellow to being blue based on conventional breeding, like the folks in Mexico have done. But then you do the same thing using genetic engineering. Is that different? Would you accept it more if it was done one way versus the other? I think people respond very strongly because of cultural values. And actually, there was a map of where these engineered crops are grown. And in the United States, we grow about 10 or more different genetically engineered crops. In Mexico, they grow genetically engineered soybeans and genetically engineered cotton, but they don't allow genetically engineered maize or corn because that's sacred to them. If you look at Diego Rivera's paintings, corn is a main sacred object. So under those kind of conditions, there's a cultural thing against having something that's been built artificially. And there's a good map right there.
That's a map.
Yeah.
It's telling us where GMOs or genetically engineered crops are accepted?
Where they're grown commercially within the legal framework of regulation. And China is a very good example where they have almost 100% genetically engineered cotton. But in terms of legally grown rice, there is 0% genetically engineered rice. And it sometimes surprises people in a top-down supposed society. The government would like to have genetic engineering, but they have a very big Greenpeace movement in China, surprises Americans, and that they don't trust that their rice would be safe if it was genetically engineered.
Josephine, do you think this tampering, tinkering, modifying nature is part of human nature? Have we been always doing it as long as there's been a recording of what we've done?
So probably. And that doesn't really tell us if it's good or bad, right? There are lots of things we have been doing for centuries, if not millennia, that are good, and some of them are also not good. Wars. So there are a lot of things that we do naturally, and they may or may not be good.
Good or bad may depend on your culture.
And it could also depend. I mean, there are a lot of some of the interesting differences, I guess, between those two different ways of getting blue corn are also to do with the time frame, right? The speed, the ability to look at something developing in
in concert with its surroundings, that you just, when you speed something up, you may or may not also miss certain aspects of it. So there are many things I think that people are objecting to when they raise concerns about genetically modified foods, and safety is maybe one of them, but there are also issues around who controls the food supply, having a limited diversity of crops ultimately, whether or not there are certain companies who then have a lot of power over things that farmers have actually been doing for centuries their own way. So there are a lot of other issues in there, I think, also around trust that are not just represented by concerns about safety.
Sam, jump up to the 70s and 80s into the modern era of genetic technology. And what were the tools that we used after we understood more about the genome? Before CRISPR, BC, before CRISPR.
So Fred just brought up, GMO, or genetically engineered foods, and the kinds of crops we have, like, corn, rice, these other ones. Those were using recombinant DNA. So, in the 70s, there was this revolution where researchers developed a set of tools that they could use to piece together bits of DNA in test tubes. So it was the first time that you could really isolate specific genes. You could package them in different ways, and you could actually introduce that genetic material into E. coli for designer bacterial strains, into plants, into animals. You could make genetically engineered mice because of this ability to manipulate DNA inside of the laboratory. Now, it's not the same precision for making changes that we might have today, but Neville, I think you were going to talk about this picture, that these are some mice that you developed yourself, huh?
This is
Is this
picture
pre-CRISPR?
This is not mice I developed myself, but these are mice that have a protein from jellyfish. So jellyfish, many species of jellyfish have this naturally produce this green fluorescent protein. And so these mice were developed to be able to visualize different cells. So we were using them to actually look to see how the axons of neurons grow during my PhD work, and find their neighbors and connect up in the brain. And so it's very nice to be able to visualize these with this green fluorescent protein. And so this is
so they glowed in the dark?
They can glow in black lights, exactly. So they're party mice, but they're very useful for science.
And how did you do it?
So this was done by a Japanese group that introduced, not in the CRISPR era, but randomly introduced this transgene. We call it a transgene because it comes originally from a different species, from the jellyfish, randomly into the mouse genome.
It sounds, doesn't sound, I read that even with CRISPR, but we're going to stay away from that, even with CRISPR, that all the discoveries have come because you've studied nature and you've looked at what nature had done, and then you build on that. So the basis of both what you're talking about and CRISPR comes from observing the way nature would move forward. Is that correct?
That's why being a bioengineer, and I mean, Sam can talk to this too, but why being a bioengineer I think is so exciting. Yeah, I mean, it's self-justifying my career choice, but it's fun because there's so much inspiration, I think, in nature, and so many useful tools that have been created over millennia of evolution on this planet. And I think the tool that we're talking about today, CRISPR and other tools, are, it's an exciting thing to do, to be able to see these working in one context in nature and then try and use them perhaps translationally for medicine or for plants.
So as we come up to today, with all the fears that people have about GMOs and the kind of crossbreedings and the things you're talking about, have any of the fears been realized? Why is the public so against GMOs?
I think probably people have different reasons that they're concerned. I do think that the safety of them just to eat hasn't been explored or explained adequately. So,
but it's been around for a long time. Right?
Well, I would say, so we have been eating genetically engineered corn in the United States since 1996. So we've had quite a few years of eating that genetically engineered corn. But the question is, how would you know if it was doing something? And I would just say, I spent quite a while chairing a committee with the National Academies looking at the safety and looking at the testing that was done. As many of you have probably seen newspapers or on the internet of these rats with big tumors and things. But looking through
From
those
GMOs?
Yeah, from GMOs. I mean, it's something that cruises around the internet all the time. But when you look at it carefully, not all the tests that have been done have been appropriate or powerful enough to say if there's a small difference. But if you take all the testing that's been done over this long period of time, there's no evidence at all that has emerged either from the actual animal testing, or even we did a comparison of people who live in the US and Canada, where GMOs are prevalent, to people who live in the UK or the EU, where they haven't been able to ask these questions about whether chronic diseases have increased faster in the US and Canada than what you see in the EU and the UK. And when you look at the numbers of things, everywhere from diabetes to cancer, different kinds of cancer,
obesity,
you don't see any change different in the US and Canada compared to the EU and the UK. Now somebody could say, well, that hasn't been a long enough period of time. But science is always that way. I think probably many of you know that if you live long enough, you find out that eggs are good or bad, or salt, you should have this much or that much. Science changes. But I think that no really strong acute changes have occurred based on people, countries where they eat GMOs.
How CRISPR came to be, the creation story, is an amazing one, made possible by the work of many labs all around the world. So my first question at this stage is, how does it work? And we're going to watch a short primer that was produced by one of the CRISPR pioneers. Jennifer Doudna is going to teach us how it works.
CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. So scientists can actually change an individual base pair in the more than 3 billion base pairs in a human cell. The fun thing about the CRISPR technology for me is that this is a project that started off as a basic science, curiosity-driven project. It was a collaboration between my lab and the lab of Emmanuel Charpentier. We teamed up to discover the function of the protein Cas9 and how it acts to disrupt viral DNA in bacteria that have a viral infection. We came to understand the way that Cas9 is programmed by RNA molecules in bacteria to recognize specific DNA sequences and then make a break in the DNA. We figured out that we could actually program Cas9 to cut any DNA sequence by redesigning the way the guide RNA in the protein is being used in cells. That understanding led to really the aha moment, realizing that this could be actually a very powerful technology in animal and plant cells. It's very exciting because it's going to enable a lot of science to be done that was impossible to do in the past.
So Sam, you worked with Jennifer Doudna, am I saying it right, in her lab. So tell us your role, but also help explain, I was confused, Cas9, is it a bacteria?
I always like to stress that there's CRISPR technology, which is what we think of when we talk about gene editing, making these genetic changes in human cells and plants, animals. But what got us all interested in CRISPR in the first place was this fundamental question she just talked about, which has nothing to do with higher organisms, but just bacteria. And thinking about how bacteria can stay healthy when they're growing in the soil, in the human gut. Every place that bacteria reside, they're actually being constantly bombarded by viruses. And these bacterial viruses are the most prevalent form of life on Earth, actually. They outnumber even the grains of sand on the planet. There are more viral particles that infect bacteria. And so there's this big question, how do bacteria protect themselves? And it's been known since the middle of the 20th century that they have innate immune systems, much like humans do. But what a number of different research groups discovered in the mid-2000s is that they have this other form of immunity called CRISPR, which uses a pair of molecular scissors, so this kind of oversimplified schematic on the slide, where they actually recognize DNA from a virus during an infection, and they slice it in half, and that leads to the viral genome or the viral genetic code being destroyed, and the bacteria can survive the infection.
So CRISPR, is that also called CRISPR?
Yeah, CRISPR has kind of become this umbrella term. Really what it refers to is the overall immune system, but the star player that's now transformed gene editing technology is a particular enzyme called Cas9. And the Cas is actually an acronym that comes from CRISPR associated.
So Cas9 is not a bacteria?
It's a bacterial enzyme, so a protein molecule, one specific protein that some bacteria produce. And actually the genesis story was done in a bacterium called Streptococcus thermophilus. You eat it every day, you have a bowl of yogurt. So it's the main workhorse to ferment milk into yogurt and other dairy products. And it was a company that was trying to make these bacterial fermentation cultures more virus resistant that led to the discovery of CRISPR actually. So every time you eat yogurt, you're eating CRISPR actually, but it was the discovery that you can take these specific pieces out of those bacteria and put them in a human cell or a plant cell or an animal cell that led to this revolution in gene editing.
To me, it's really fascinating. I think you mentioned this, but it's worth stressing this, that this is an adaptive immune system in bacteria. Bacteria are tiny single-celled organisms. You know, we're these, trillions of interacting cells that make us up, and we have an adaptive immune system. We know we have antibodies and we have T cells that fight off infections, but that's because we have so many cells. But to me, it's just mind-blowing still that a single cell, a tiny one-celled organism, has an adaptive immune system, meaning that word adaptive means it can have a memory of previous invaders, previous viruses that it's encountered, which it keeps in this CRISPR area of its genome, and then when it sees something that matches that memory, as Sam said, it's a molecular scissors. It can cut it and get rid of it before it harms and sickens the bacteria. It's very cool.
Let's do a round. We'll start with Neville, come all the way around. Tell us in your own, and from your own perspective, in your own words, what you think is so revolutionary about CRISPR. Neville, you start.
Oh, okay. That's a good question. What's so revolutionary about CRISPR? Yeah, I mean, it's kind of hard, I think, to overstate that what it's enabled us to do. So basically, one thing that we as biologists and geneticists have been limited in some ways is we have this concept in biology of a genetic model organism. So you might hear a lot of studies that involve either bacteria or fruit flies or mice. And you might wonder, well, why do scientists just love these couple organisms that I keep hearing about? Why do I hear so much about mice but not about rats? Is it just that mice are cuter and scientists like using cute animals? There's a reason actually that they've kind of fixated on a few different organisms, and that's because those are genetic model organisms, meaning their genomes have been easier to manipulate so we can understand what changes in their DNA, in their genetic code, what they do to those animals. We can understand the connection between the DNA, the genotype, and the phenotype, how it looks to us, how the animal behaves. And that's been the case up until very, very recently. And I think one of the real breakthroughs in the CRISPR era is the idea of the genetic model organism is very different now, because everything can be a genetic model organism, including what a lot of people consider the most important organism or model organism, which is us. We'd like to study something to understand about how our genes result in maybe diseases that we get, and how we can repair those defects. And so human cells, the DNA was actually quite hard to manipulate until recently. And really, any organism now is a genetic model organism with CRISPR.
I'm supposed to follow that?
Yeah, try to follow.
I went on too long.
Can I just pass and say that too?
I agree with that.
I guess for me, I mean, I love your wondering about how does this work, because that's what's gotten me excited, is really the mechanics of the whole process. And I think what is often unappreciated about even CRISPR is that it's become a moving target, because there's no one thing that defines CRISPR, because what we've discovered over the last 10 years or so is that there are all different flavors of CRISPR where the Cas9 protein that we've been talking about isn't even present, where different strains or different groups of bacteria have evolved different types of enzymes that might work similarly but do completely different things, or not even edit DNA but edit RNA. And one thing that a lot of researchers are doing now is continuing to harness nature and kind of remain inspired by what bacteria and other microorganisms have evolved to be able to do over evolutionary time, and then think about how can we re-engineer the system and use it in more and more creative ways to solve problems.
Okay, good.
I'm
it's
up
like
next.
a contest.
All right.
Isn't it?
Yeah. Right. Okay. So here we go. So we'll take this to a higher level. Okay. All right. So I'm an
Ooh, that was pretty high, Fred.
Well, I'm an entomologist. All right,
Fred,
I'm in an entomology
can you bring
department.
it to a lower level, please?
Right. So I study insects, and we're concerned specifically about agricultural, insects, pests on crops, but also mosquitoes that transmit diseases. And one of the most talked about is malaria. And when we think about using CRISPR to decrease disease, sometimes we're thinking about manipulating humans so they won't get the disease and so on. But with something like malaria that is transmitted by a mosquito, people have been working for many years with transgenic mosquitoes where you've changed the genes to make it so the mosquito can't transmit malaria. Now, it sounds really good, but think about it. If you release a couple of thousand mosquitoes that you've bred in the lab, there are going to be millions of other mosquitoes that do transmit the disease. So what we've been working towards for a long time is a way to get those genes to move into the population based on being inherited more than expectations by Mendelian inheritance, so that it'll push the gene into the population. And people have worked for a long time on how to do that. And what has happened with this CRISPR technology that came from the biomedical researchers was this recognition that what they're trying to do is put the gene into an organism and have it function. But that's just a one-off thing. And the idea is that once you put that gene in, you can use different manipulations so that it automatically continues to put itself into new organisms. So when you give birth, all of your offspring will have those genes instead of half of them. So if you think about linking that with a gene for not transmitting malaria, all of a sudden that mosquito population becomes a nuisance, but not something that's transmitting malaria. So that's exciting.
That's really exciting.
In my lifetime, this is the first time I've had in my lifetime that we've actually seemed to have the means not just to select the genes of future generations, but to actually change them. And so that's monumental prospect. It isn't anything someone's doing right now that we know about, but it's certainly like dangling there as a much more real prospect. So, decades of people thinking about being able to do something like that to actually potentially being able to do that is monumental. And the other thing that's come along with that, I think, was this really, I think, brave and really important move that Jennifer Doudna and other people involved in the development of this technology made, which was immediately asking for an international conversation about that potential. So that moment of open science and raising questions about your own work and acknowledging that you, the scientists, are looking for everyone else to join in the conversation, that's a monumental moment as well.
I want to move us, though, into the whole question about genetic changes in the human body and medicine. Mm-hmm. And so let's divide this up. First, let's talk about non-heritable diseases, and then we'll talk about heritable. So non-heritable diseases, some cancers, some blindness. What benefits does CRISPR offer over the earlier technologies in this whole area of non-heritable? Sam or Neville, either one of you?
Sure. I don't know if I'll address the non-heritable part exactly, but I think the key, we talked about this earlier, but really the key property has been the easy programmability, that this is really getting into the realm of almost like programming a computer. It's something that we can do, except we do it in the lab, but quite easily. And so it's enabled us to ask questions really of a different scale. So in previous decades, you'd often have a geneticist who spent their entire career working really just closely focusing on one gene and really exploring what happens, where that gene is maybe mutated or has other changes to it, and or what patients with conditions that involve that gene look like. But here, with this easy programmability, we can now, a single investigator, a single scientist can actually ask really genome-wide questions. They can say, of all 20,000 genes in the genome, that's how many genes are in the human genome, what are the effects of each of these genes on the growth of a cancer cell or on resistance to a cancer therapy? And so that's some of the work that we've been doing in my lab.
And you can ask all the genes at the same time?
All the genes at the same time, a single scientist. And that's really an unprecedented scale where we can develop libraries of CRISPRs, each with a different guide RNA, this component that Sam mentioned that programs the Cas9 or other CRISPR enzyme to go to different places in the genome. A library of guide RNAs that target all of the 20,000 genes in the genome. And then we can have this pool of cells, and this is, again, human cells. This is the model organism we care about in biomedical disease science. And have this pool of cells and then say, which of these cells now are resistant to a drug that they shouldn't be? And okay, maybe this gene, if we see a patient with a mutation in this gene, we now know we shouldn't give them this drug. We can predict in advance of when they come into the clinic that this might trigger resistance. These are the kind of, we're not quite there yet, but this is where this is heading.
Let's say I have cancer, and we're at the stage where CRISPR is ready to help me. So how do you administer, or how will we administer CRISPR changed medicines?
Yeah,
is it IV? Is it?
So I think it's impossible to predict the future in this field is changing rapidly, but I think we have a clue by the clinical trials that are just being registered right now, and a lot of them involve taking advantage of the immune system. That's been a huge transformation in cancer therapy over the last decade, is the introduction of immunotherapy first in clinical trials and now in many FDA-approved medicines that encourage the immune system to fight cancer. And so instead of administering drugs or antibodies on a continual basis, there's the idea of taking out a small portion of cells, using gene editing to modify those immune cells, and then having something that would not require continuous administration. Maybe it would be administered just once. These are the trials that are starting now with gene
humans.
editing.
On humans.
On humans.
Maybe I could talk about one example. It's actually, as far as I understand, the first clinical trial using gene editing in the pre-CRISPR days, so it's a different platform, but same basic concept. For HIV-positive patients, and the way this clinical trial was administered is patients have immune cells removed from their blood. They're then edited at a particular gene that's been known for decades to confer resistance to HIV viral infection. Mm-hmm. So the idea is you actually take a patient's own cells, you transform them into cells that can no longer be infected by the virus, but they're otherwise still the patient's own cells. So it's not coming from a donor where you have to worry about immunocompatibility. You edit the patient's own cells, and then you deliver them back into the patient's bloodstream, where hopefully those cells will take hold and now generate a new reservoir of the patient's edited cells that have HIV resistance, and will pass on that resistance to all cells that end up coming from cell divisions. And if you can hit the stem cells, even better, because now they're going to have an immortalized, permanent line of cells in their body that have been edited and can never be infected by HIV.
And what's amazing about this is there's actually people that carry this mutation naturally. There's a few very, very lucky individuals that are basically immune to HIV, and it's through studying the basic disease mechanisms of those kinds of individuals that we now know what gene could be targeted in the rest of us to have HIV immune cells.
This technology has been offered in an open source way. So it is available for scientists, am I wrong about this, all around the world, any lab, unscrupulous people or not. It's even offered on the web. You can buy a CRISPR kit today online. So, and is the horse out of the barn, or can we get something, at least a discussion, a serious way to make sure that things about what you're talking about doesn't happen, to make sure some guy isn't making a monster in the basement, all kinds of possibilities. And it seems that we're there, and we haven't talked about, we should have talked about it already. We should have been regulating it already. Why is it open source, for example?
I can only say something about the open source because I think the rest of the question is very important too. But I think in terms of science, there's some examples where things were a little bit less open source. Today, it's much easier to share because of things like the internet. But previous technologies, one famous one is PCR, polymerase chain reaction, which is a very famous technology in molecular biology, won a Nobel Prize. It allows us to very easily amplify specific pieces of DNA. It was created in the mid-'80s. Very powerful technology that was initially patented by the company that discovered it and kept in a quite proprietary way. Now it's widely disseminated. I think something that's different about the CRISPR community, and Sam's been a key part of this too, is that all the labs, lab that I came from, Feng Zhang's lab, was really pushed to the forefront, the idea of very quickly putting these tools in a nonprofit repository called AdGene that would distribute them to any scientist for a very nominal cost. And to think, there's some other things you can focus on too, but one thing that's really important to bring up is this is unusual in science, how quickly this has been shared, and it's had an unbelievably positive impact on allowing biomedical scientists to just do a lot more and a lot faster than they could get there.
But who's thinking of the downside?
So that's I'm going to talk about the upside.
Okay.
Maybe you can have somebody else, but that's been my experience, and it's really been different than a lot of other science, and it's hard to overstate that actually. Yeah.
None of the scientists want to talk about the downside.
I can say a little bit about the uses in humans, right, and in embryos and sperm and eggs and things like that. But I don't know the answer to these questions about how it stops somebody from ordering it over the internet and using it in an animal or an insect in a nefarious way. But certainly when it comes to humans, so even in the US does not have, comparatively in an international sense, doesn't have a lot of federal regulation that deals with in vitro fertilization or these reproductive technologies. Other countries, some other countries like the UK and Canada and Australia and various other countries have sort of legislation that regulates and oversees manipulation of sperm, eggs, and embryos, and makes rules about what can and can't be done. And a lot of those countries have laws that say that you cannot make heritable changes to human embryos, for instance, or to the eggs or sperm. So can't make a change to the genes that will be passed on. Now they might be reconsidering just exactly how stringently they want to stick to that.
Even if it's positive.
So at this point, yeah. So that's. But just to say that, in 1990, the UK developed a regulatory scheme and an oversight system for looking at all of this. And right now in the US, there is a little bit of data collected from IVF clinics so that we have some idea of what's going on in terms of like how many cycles of IVF are there and how do they use donor eggs. Any potential use of these gene editing technologies to make a baby would have to go through the FDA.
What about saying you can't make a change, in the embryo?
I think that's something that needs to be discussed, and it is being discussed. And Josephine earlier mentioned, Jennifer Doudna and a very large group of scientists, but also non-scientists, importantly, have met. 2015 there was an international meeting. I think there's another one planned for the end of this year. And I think that's a critical piece to really having a broad discussion about what, on a ethical and social level should and shouldn't be offered. There's scientific technicalities about, off-target effects and unintended consequences, but I think there's also a bigger question, which is what kind of society do we want to live in and what types of interventions should be supported when, frankly, there's a lot of other social problems in our society that would be addressed with a lot less money than might be put into, generating genetically edited babies for a very select few individuals. So I think it's,
but
whether or not a
discussing it. Your community is out having conferences to discuss the ethics, and what regulation makes sense?
I mean, I think it's your community.
Yeah.
I mean, the National Academy of Sciences of the US did a whole report on the prospect of using gene editing in humans, including to make changes that could be passed on from one generation to the next. And in that report, they argued for allowing potentially some heritable changes, but if they met certain really strict criteria. So for sort of stepping a wee bit back from the idea that we can't make a heritable change and saying, well, maybe we can if we can show that it's safe and effective, and if it's a disease that's really serious versus, like, you know, giving people purple eyes or something. So like there is a discussion, and it's not just amongst scientists.
Think about a heritable disease that you could eliminate from the face of the earth. I mean, that's possible. That's part of this. And why would anybody
Or
try
let's
to
say those
stop
rare
that?
cases where you have parents that have no other option to have their own child that will be free of a disease. No intervention, as many cycles of IVF and genetic screening of embryos, none of that will allow them to have a child that doesn't inherit their disease.
Yeah.
You could make a strong argument for if there's a technology that can address that problem and we can administer it in a socially responsible way, should we have some red line that says, no, if you change the DNA of an embryo, that's wrong, but if you do it in a patient that lives with the disease, that's okay. I mean, some people might say, why are we making this distinction? We would be okay intervening surgically on a newborn baby. What's the issue with doing it earlier in development if it's a genetic intervention?
And actually that's why the UK recently changed their law around this issue to allow for something called mitochondrial replacement technology, which isn't CRISPR, but it does introduce a heritable change into the embryo. And so they had a whole public debate, they debated it in Parliament, and they made a change to their law to allow for this. So I do think that there are ways in which regulatory structures can be adaptive to these really compelling cases, without necessarily just opening the gates.
I'd like to go back to the comment that two of you made about getting rid of these diseases, this idea that if you had the ability to get rid of the scourge, and that sounds like it's global. And I guess even if these technologies become pretty feasible, and I think Sam was bringing this up, who is going to get to use them? You know, are we really going to be able to eliminate a disease like that requires us to mess around with an embryo, even if we're great at it, when we can't even give people enough to eat, right? So who's going to get it and who's not going to get it? And that becomes a real important social issue as to, you know, where are you going to go with this?
I salute you all. I hope that we didn't talk over your head or under. I hope we hit it just right. That was our goal. So I want to thank you all for the work you do, progress you've made. Well, thank everybody.
Thank you.
Thank you. Thank you.
On April 4th, 2017, a privileged group of telescopes on mountains across the planet switched on at the same time. For the next week, they danced in unison, collecting radio waves dispatched from the center of our Milky Way galaxy and from the galaxy M87. Together, they make up the Event Horizon Telescope, a global project to capture the first ever picture of a black hole. That's right, ever since physicists first conceived of black holes centuries ago. Every image of one from our textbooks and our space agencies, they're all illustrations. Until now.
We are delighted to be able to report to you today that we have seen what we thought was unseeable.
For centuries, physicists have theorized that an object with enough mass and density could trap even light in its gravitational field. Just as you have to travel faster to leave Earth than you do to leave the moon, there could be a place where you'd have to travel faster than the speed of light to escape, and nothing moves faster than light. The math from Einstein's theory of general relativity describes an area completely invisible to us within a boundary called the event horizon. And at the center of that black hole is a singularity, a point of infinite density, which is where physics as we know it breaks down.
They showed up in the math long ago and they kept reappearing and they sort of persistently would not go away. But Einstein always thought that there must be some physical mechanism that prevents stars from collapsing to an infinitely small point, which is actually pretty reasonable. I mean, because it sounds insane.
Eventually, scientists began to see things that only made sense if black holes were real, like the orbits of these stars around the center of the Milky Way galaxy.
You see these stars just slingshotting around an invisible point. And a black hole is the most likely explanation for putting that amount of mass in that small space for something that's completely dark.
We can also see the glowing material that spirals around black holes. Friction heats this matter up tens of millions of degrees, and anything that hot emits X-rays that we can detect with telescopes that orbit above Earth's atmosphere. This is a pair of galaxies that pass through each other. There are at least 9 suspected black holes here, but you can only see them when you look at the X-ray layer. These dots are X-ray sources linked to suspected supermassive black holes at the center of galaxies 3 to 10 billion light-years away, and that's just from this small patch of sky. Some supermassive black holes also feature gigantic jets of particles, seen here in radio wave data from the galaxy M87, which has a much bigger black hole than the one in the center of the Milky Way. No other known source of energy could power these things, and nothing we know of besides 2 black holes colliding could have produced the gravitational waves we detected in 2015. Scientists think there are black holes, large and small, all over the universe. We can see their fingerprints, but we didn't have the mugshot. Directly imaging a black hole has been impossible because they're either too small, too far away, or both. Sagittarius A star, the black hole at the center of our galaxy, has the mass of 4 million suns, but it would fit inside the orbit of Mercury. Imaging it from Earth is like taking a picture of a DVD on the surface of the moon, with huge clouds of dust and gas in between.
So many things had to go right for this image to exist. So the first thing that has to happen is there has to be some slice of light that travels all the way from the edge of the black hole without getting knocked off course or absorbed by any of the gas or anything in between. And then it also has to make it through the Earth's atmosphere, which a lot of frequencies of light don't.
They landed on a wavelength of 1.3 millimeters at the high-frequency end of the radio spectrum. With that wavelength and with 8 observatories across the world, the Event Horizon Telescope had a chance at seeing a black hole, as long as the weather cooperated.
You have to have clear weather in all of those places at a time when the Earth is oriented in such a way that all of those telescopes can see the black hole simultaneously. They can really only observe once a year.
There was so much data involved that it had to be flown on airplanes. They waited 6 months for the hard drives to arrive from the South Pole, which closes during wintertime. This multi-telescope method is called Very Long Baseline Interferometry. It correlates time-stamped data from distant telescopes to boost the signal and quiet the noise. Each pairing of telescopes contributes a piece of the puzzle. But the image doesn't just pop out after that. They had 4 groups working for months to generate the image that best represents the data.
Each group was working individually and in isolation from the other groups, working with the same data to see that each group came up with the same image or not.
Here it is.
And the result of all that work is this. The bright parts are the matter and light swirling around the black hole, and it's brighter on the side that's moving toward us. And the dark part is the black hole's shadow, which includes the event horizon plus a region where light could escape but doesn't. The size and shape of the shadow appear to confirm the theory of general relativity.
Today, general relativity has passed another crucial test, this one spanning from horizons to the stars.
Humanity's first image of a black hole isn't crisp and beautiful like the illustrations or the movie Interstellar. It's better.
The picture we see this week is made of scraps and bits of light that's been traveling across the universe and collected by these, aluminum dishes on top of mountaintops and then combined in a supercomputer to make this image. So that's why it's real.
Talking about black holes, of course, Stephen Hawking is the great scientist who comes to mind. So I'd like to spend a little bit of time thinking about what it is that Hawking told us about black holes, what were the insights that he gave us, and what puzzles emerged from his work that we are still struggling to resolve and probably will continue to struggle with for some time. And toward that end, as a little bit of background, we need to have some understanding of a concept that everybody is familiar with at some level or another, but let's use this opportunity to get us all in the same place, and that's the concept of entropy. So entropy is a word that's freely used in the culture. We often use it to describe a certain amount of going from order to disorder. That's going from low entropy to high entropy. That's sort of the intuition that the culture has about this word. So let's start with that very basic way of thinking about entropy. And a good example would be, let's say I take my book, I throw it up in the air, it starts very ordered, all the pages were in complete numerical order, but as they come down, it's overwhelmingly likely that they will not land in numerical order. They'll land in a highly disordered state. And that gives you some sense of what it means to go from an ordered arrangement to a highly disordered arrangement, going from low entropy to higher entropy. Now, of course, that's just a sort of fanciful everyday example. If we head more toward a physics example, we could imagine having a box of gas. And imagine it just has a small number of atoms in there. And imagine that they're all neatly arranged in this nice cubic lattice. This would be a very ordered or low entropy configuration of the gas, like the pages all in numerical order. Now contrast that with a box of gas that has a whole lot of atoms, a whole lot of particles are all bouncing around in a completely haphazard way. This would be a very disordered state, a high entropy state. And another way of thinking about the distinction between low entropy and high entropy, which we'll be making use of in just a moment, is the amount of information that would be required to describe these two configurations. Because look, on the left, you don't need a lot of data. It's a cubic lattice. You've got 3 by 3 by 3 atoms filling out that lattice. And if I give you the separation of them, say 10 centimeters, that basically describes the configuration on the left. You don't need a lot of information to describe a system that has low entropy. But let's say I wanted to describe the configuration on the right. Well, I'd basically have to tell you where each and every particle is located. That's the only way I could describe it. And look at all the information I need to describe that configuration. So the idea is low entropy, not a lot of information. High entropy takes a lot of information that's hidden in this configuration of particles. And that's kind of the distinction between order, disorder, low entropy, high entropy, low information, high information. Now, of course, I'm talking in loose language so that we can all get a feel for the critical ideas, but you can make this all quite precise. And this gentleman over here, you know who this is? Who's this? Ludwig Boltzmann, that's right. And this is his famous tombstone, and you see that formula on the top of his tombstone, and that is the formula that makes these statements precise. This is a formula that you learn if you take a course in statistical mechanics, thermodynamics. S stands for entropy, K is Boltzmann's constant, and the rest of it has to do with the number of configuration of the ingredients of the particles that make up the system. And the famous second law of thermodynamics makes use of this formula, and again, it's something that we're quite familiar with in the culture, that things typically go, as I said, from low entropy to high. That's the natural progression of a system, which means that this quantity S tends to increase over time. The entropy later on is almost always greater than the entropy earlier on in the evolution of a system. And what we'd like to do now is have this idea come into contact with the idea of black holes. And we'll see that there's a certain kind of tension that arises. It's a very fruitful tension that will take our understanding further as we try to come to grips with it. And to help us go on that journey, we're going to now bring out the Donner Professor of Science in the Department of Physics at Harvard, whose primary area of research is string theory. He's received numerous awards and recognitions for his work, including the Dirac Medal and the Breakthrough Prize. Please welcome Kumran Vafa. Now, I should say that I have known this gentleman for over 30 years. We were, I guess, both postdocs at Harvard in the 1980s.
That's right.
You got a job at Harvard as faculty, but that's okay. We're still good.
You're doing very well too.
Yeah, thank you. And I should say, my most exciting intellectual adventures in string theory have been the papers that we have done together.
You.
So
we had a lot of fun with you, Brian.
Thank you. So it's been an honor to work with you. So thank you. So let's now get into this subject here. We heard a little bit about entropy and about the second law that entropy increases over time. But in the 1970s, people like Bekenstein and Hawking, and they began to worry about how the second law of thermodynamics would behave in the environment of a black hole. So you can give us a sense of what they were puzzling about.
So part of the puzzle was the objects they knew falls into the black holes, but they cannot come out. So objects that are falling in have entropy, just like what Brian already described. You have some information about where they are or how they are, but once you fall into the black hole, then you don't have any more access to that information. So what happened to that information? So that seems like you lost entropy. Entropy went down, and that's against what Boltzmann said.
So can I give a concrete example of that? So I think we have a little silly example. If I took my wallet, it has information, it has entropy, I throw it into the black hole, and you're asking what happened to that entropy?
Exactly.
Did it disappear from the universe?
Exactly. So that was the kind of question that they were struggling to understand. How could this be consistent?
Right.
So they began thinking about this, and then there were a few ideas that they were thinking about, and in particular the properties of the horizon of a black hole.
The edge.
The edge, the event horizon that we talk, the edge in which, beyond which no light can escape. They found it had some interesting properties, and in particular they began studying what is the area of this edge, and how does it change? And in particular, what happens if you throw objects into this black hole? What happens to this area? And one of the surprising things they found was that when you throw objects in, this area cannot stay put, and in fact increases. So they began to think about, okay, what could this mean about this, what this could be related in some way, compensated by the fact that you're losing something, the entropy. Could it be somehow compensated by the fact that this area is growing? So they begin to ask the question, could the area of this event horizon be in any way related to the lost entropy?
So we actually can unpack that. So let's just go through a couple of little examples here. So this is the standard second law of thermodynamics, that entropy grows over time. Let's just take the two boxes that I started with to illustrate what you're describing. If we take those two boxes and we put them together, what happens to the entropy? It goes up,
That's
right?
the Boltzmann's statement.
Right. So we have the combined entropy is greater than the individual's entropy. Now you're describing something that doesn't have to do with gas in a box. You're talking about the geometry of a black hole.
So for example, they studied what happens if you study one black hole with one area of event horizon and another black hole with a different area, and you bring them together, you merge them, and you get a new black hole with a new area. And they found that the area of the new formed black hole is bigger than the sum of the two areas. Just like the entropy had that property. The entropy resulting in the box when you combine them was bigger than the individual entropies. So the entropy sounded very similar to the area, or area sounded very similar to the entropy. So they began asking, could it be that the area and the entropy are proportional to one another for black holes?
And indeed, they found a formula that we can bring up here. So entropy is the area, and the proportionality constant is a little bit complicated.
Kc over 4gh
right, but you don't really have to worry too much about this number on the right-hand side. But the basic lesson is that they're guessing, they're suggesting that the area of the event horizon is somehow holding on to the entropy, holding on to the information in some sense that's inside the black hole. Now, you gave an interesting thought experiment about how to test that. You were talking about throwing something in and watching the area. And it just so happens that we have a little segment on that here. So why don't we go through this? So here's so what are we seeing here? And then we'll do your little thought experiment.
So here, I guess we are seeing a black hole, a red black hole.
Yes, exactly.
With the event horizon of it, you can see that the idea that the information is proportional to the area with some proportionality constant, if each individual element of the information is like one square there, we just divided this sphere into these areas, so these represent bits of information on that surface of that black hole.
So those little squares can, roughly speaking, be thought of as Planck areas, using the Planck length that describes sort of the smallest length that makes sense in a quantum theory of gravity.
Squares of the size 10 to the minus 33 centimeters or so, if you want to know.
10 to the minus 33 squared. Yep.
Each size of the square is.
Yep.
Yeah.
So you basically count the number of those little squares that are on the event horizon, and that's the amount of entropy it has. So if we now if this is true, and we were to take a particle that sort of carried 1 unit of entropy, what would we expect to happen if this is true?
We expect this area to grow exactly by 1 square.
So let's sort of do that. We have, this is actually a real film of space here. So let's get our particle going in to this guy over here. Good. So the one on the left was the original black hole. The one on the right is the larger one after a particle has fallen in. So if we take all those little squares and we smear them out on the larger black hole, what we'd expect to happen is something like that.
Exactly. So one more square.
So we throw sort of one particle in that carries 1 unit of disorder, and the black hole eats it. It grows a little bit, and in fact, you can calculate that the amount by which it will expand can accommodate 1 more of these little tiles, and that makes this a consistent way of thinking about black holes. So this was like an important, how important was this kind of an insight?
Well, it was the amazing insight because it was the first time where Einstein's theory of relativity was combined with quantum effects. And that is what Hawking used to actually argue that the entropy of a black hole has something to do with the area. And so this combination of quantum theory and Einstein's theory was quite novel, and this was in early 70s, mid-70s.
So is this the end of the story when it comes to entropy and black holes? Like, what issues does this leave over?
Well, actually, the problem was that Hawking's argument showed that there must be information, but on the other hand, the horizon of a black hole is featureless. So you don't see these point squares or anything. So there seems to be no room for information to be stored on it. So, in fact, if you use Einstein's equations and solve it, you find there's exactly one solution for a spherically symmetric black hole. One solution means it has zero entropy. It's exactly ordered state. There's no choice.
One piece of information.
One piece of information. And that is inconsistent with what Hawking found when he combined Einstein's theory with quantum mechanics, where he found a huge amount of information. And the question is, where is this huge amount of information stored? Is it the shape of the horizon, or is it what aspect of black hole encodes the microscopic structure of the black hole? That was open when Hawking discovered this fact.
So for Hawking's idea to work, let's just go back to my little example of sort of throwing the wallet in. So in that little example, I threw the wallet in and we said, where did the information go? So the new idea, if it's true, would be I take my wallet and I throw it in, and rather than it like disappearing, the idea, roughly speaking, would be the information that is in my wallet, the configuration of all the atoms and molecules, it goes toward the black hole. And we want it to be the case that that information kind of like smears itself out on the outside and is encoded in sort of bits, ones and zeros.
That's the picture.
That's the picture. And in principle, were this true, I could actually extract that information in principle and use it to rebuild anything that fell in this case, it would be my wallet. So that's the idea.
That's if the information is not lost and information is there. That's what we would expect.
Right.
Yes. That's the hope.
And the puzzle is that actually...
Is that actually true? And how do you actually see this information? Or where is it stored? Or how do you account for the microstates of the black hole, or the entropy of the black hole?
And that's where you come in.
Well, me and my colleagues in string theory began studying these questions more seriously with the advent of duality symmetries in string theory. So first
string theory, can you say a few words on string theory?
I should say a few words about string theory. So string theory is a framework where we believe combines Einstein's theory of relativity with quantum theory in a consistent framework. And as part of it, it demands that the entities making up the matter are not just point particles, but entities like strings or membranes or extended objects have to play a key role. And so that's one aspect of string theory.
So here's a little, if you want to take us through, this is just a little example of what string theory might look like. So you got a piece of matter, and we're going to dive into it, and then you're saying that if we go sufficiently deep...
So if you go to the beginning inside, you'll see the atoms first, and inside the atoms you see the nucleus, and the nucleus you see, well, here I guess the protons and neutrons, I'm assuming. And you go inside them, you see the quarks, and once you see the quarks, you can go inside them, and if you zoom in a little further, you might see actually that these featureless quarks that you think are point particle actually are tiny strings, vibrating strings of some sort. And that's the kind of a picture that we expect to be true in the context of string theory, that point particles are point-like because we are not zoomed in sufficiently close to them. And if you zoom in enough, they will have features like strings.
How big would those?
We don't know exactly, but it could be like 10 to the minus 30 centimeters, somewhat close to the Planck scale that we discussed already in the context of the areas of the horizon of the black hole. So that's the basic feature of string theory, but as one of the aspects of string theory that was originally one of the negative features was that string theory predicted that the spatial dimension of the universe is not 3. And that was manifestly in contradiction with the fact that we think we live in 3 spatial dimensions. We have 3 spatial dimensions. String theory seemed to demand that we have 9 spatial dimensions. And so this was a contradiction originally, and physicists thought, how could this possibly work? And one resolution was, these 6 extra dimensions that we have are so tiny, like little circles at every point in space. Let's imagine 6 circles at each point, 6 types of circles, which are so tiny that you cannot see. So the idea would be the macroscopic dimensions are only 3. The big ones are 3, but the tiny ones are 6. And the tiny ones are hard to see unless you really zoom in, and that's difficult to do with the energies that's available to us in our experiments today.
But the idea would be that you've got these big dimensions that we know about, these extra dimensions, And then how do you relate this to this idea of black holes?
So this was actually a negative feature for string theory. So string theorists were kind of struggling. Yeah, we have these 6 extra dimensions. It's a little embarrassing. It's tiny. You don't see them. And then other people said, sure, yes. So this was the state of the art till mid-90s, where we didn't know what to do with these extra dimensions. They were there just to make the theory consistent, and we were hiding it away in tiny little things. And then came the question about the black holes, and the black hole had the opposite problem. The problem with the black hole was we were missing information of the black holes. Where were they? What were that information? It turned out that these two problems canceled each other out. And so the problem that was where are the ingredients or the degrees of freedom that constitutes black hole ended up in the internal spatial, tiny spatial things that I was talking about. So as you can see here, the red donut-shaped object there is to represent these tiny internal dimensions, and the blue sheet that you see represents the macroscopic, 3 macroscopic dimensions. And so the idea there is that if you take a string or a membrane or one of these extended objects and wrap it around the cycles on these external tiny spaces like a torus or like a circle or donut or whatnot shape you have, it will create a mass at the point where you're wrapping around that circle. And the more it wraps around, the more mass is going to be constituting there, and the more it's going to shape and distort the geometry of space around it. And if it's enough of it, it's going to create this warped space, which is what we call the black hole. So the black hole itself can be viewed as these strings or membranes wrapped around these extra cycles of this tiny space. And so then if the question is, what accounts for the degrees of freedom of the black hole? These are precisely these strings or membranes wrapped around these extra dimensions. How many are there? How many degrees of freedom are there? What are the entropy? Gets translated to how many ways these extra strings or membranes can wrap around these extra dimensions. And there are many ways. And that accounted for the entropy of the black hole. So with Andy Strominger and I did actually a computation of how much entropy is there in these wrappings of these strings around these internal dimensions of string theory, and we found exact match with the prediction that Bekenstein and Hawking had made about the entropy of these black holes.
So just to quickly unpack a little bit, so basically you found a new way of describing black holes in string theory, which makes use of the extra curled up dimensions, the red part of this image that we find here. And you're saying that by wrapping these strings or ingredients of string theory around the extra dimensions, you can create a warping in space that looks just like the black hole that we've been talking about for many years before string theory even was on anybody's mind. But in this description, you can do a direct count of the amount of information necessary to describe the situation, and bang on, it matches what Hawking said.
Yes.
So this gives us insight into one possible way of describing the internal structure of a black hole, which in many ways I think the community views as one of the most important pieces of data that string theory may be going in the right direction. We don't have experimental support for these ideas, but here's a mathematical piece of experimental support, if you will, that it matches ideas that have been on the table for 25, 30 years. So this is a key step forward. But now let's turn to some of the puzzles that still remain. And part of the puzzles surround something that's tightly related to what we've been talking about, which is something called Hawking radiation. Can you just tell us a bit about what Hawking radiation is?
Yes. So Hawking, after discovering the properties of having information in the black hole and the entropy, he also noticed that the black holes actually are not quite black. And what happens, if you go near the event horizon of the black hole, what happens is that you get pairs of particles created out of vacuum. These are quantum fluctuations. The quantum fluctuation is always creating particles and antiparticles in pairs, and typically they go in and out without doing anything. But if they are near the event horizon, one of them perchance could go towards the inside of the black hole, and then one of them could go outside, and the one which goes inside has no way of getting out, so it's stuck there. But the one going out, so it can leave and go far away from it, all the way to infinity. And that looks like a radiation from the viewpoint of somebody outside. So in other words, from the viewpoint of somebody outside, the black hole is actually radiating energy. As it radiates energy, it loses mass, and it shrinks and shrinks and shrinks, until after a while, the black hole totally disappears, where there's no more energy left to emit. So once the black hole disappears, the question is, what happens to all that information that went into the black hole? What happened to that area I was talking about? Where is that information gone? So this was a question that Hawking basically posed. Perhaps you recall the discussion between Einstein and Bohr, that Einstein did not like the probabilistic aspect of quantum mechanics, and so he said, God does not play dice. And Niels Bohr responded by saying to Einstein, stop telling God what to do. And Hawking added his own wrinkle on this. He said, well, what happens if you throw a dice inside the black hole? And the black hole after a while disappears, and with it disappears the dice. So, and then he said, not only does God throw dice, he sometimes throws them where they cannot be seen.
And then you made a little cake for him.
So yeah, we had the lucky occasion of having Stephen Hawking as our guest at our home a couple of years ago when he was visiting Harvard, and we made a cake in the form of a black hole with the dice, and the book, his famous book, which is actually made of, is a cake, and it's actually gluten-free because he was allergic. So Stephen had his cake and he could eat it too.
Very nice.
So in that picture, you see that there are two dice hanging there, floating towards the black hole, and it's going in. And that's the issue of the information puzzle. You lose the information of what is the dice roll, and that's what's called the information puzzle, because if the dice rolls some number and the black hole evaporates and disappears, with it disappears the information about what the roll of the dice was, and that's called the information puzzle, which we still are struggling to understand even today.
Now, most people in the field for a long time have anticipated that somehow the information does get out with that radiation that we saw coming off of the black hole. Now, for a while, Stephen Hawking said that the information would not come out. I mean, that quote was quite serious. He was saying that we have to rethink quantum mechanics because the information does not come back out. He then changed his mind on that.
Yes. So part of the reason for this is the discovery of a more detailed version of what is called holography. In other words, this began with the work already back in the long time ago, but it became more precise with the work of Juan Maldacena, where it was discovered that you can describe a gravitational system precisely using objects which are much simpler to understand. And in particular, in that system, you could prove mathematically rigorously there's no way you can lose information. So therefore, indirectly, there was a proof that whatever happens to black holes, the information should get out. So that part was kind of beginning to be established using these kind of arguments in string theory. But the wrinkle is that even though there's this mathematical proof that this should happen, we still did not know exactly how. And some people thought maybe the information gets out with this radiation, as you were saying, Brian, very gradually and very tiny bit at a time, but gradually builds up to give you information out. And this is what in the early 2000s, I believe, Stephen agreed that this is the resolution. But I still think, and I think the community still believes, we haven't really understood how this happens exactly. So the information puzzle about how this information comes out is still one of the big mysteries of black holes, and we don't really understand it.
So do you think it's possible that we will one day conclude that the original view of Hawking was right, that the information does not get out? Is that still on the table in your view?
Well, I think it sounds much less likely given that we think we understand a dual description, another description of these black holes. So it seems like that seems much less likely now, but you know, we can never say we know everything about the subject. That's our current understanding. It could, of course, evolve. But even with that understanding, our understanding is not sufficiently detailed to convince us of how it happens.
Now, a lot of our discussion here has been focusing on what happens at the edge of a black hole, at the event horizon of a black hole, that it may be where the information is stored, it may be where the entropy is stored. We haven't said much about what happens at the center.
Right.
Can we just spend a little time talking about that?
Well, one of the things that happens in the center of the black hole is that there's this singularity, this infinity that Brian discussed already, this division by zero analogy. And we don't believe that's physical. We believe that's just a phantom of our equation. It's just a mathematical thing that comes out, and that means that our description breaks down, not that there is an infinity.
Now, quickly, the infinity could be physically that you've got all this mass crushed to zero size.
And
so
zero size is not physical.
Yeah.
For us, there's nothing smaller than Planck's length, which is about 10 to the minus 33 centimeters. The notion of space breaks down anything smaller than that. So there cannot be an infinity in physics. We don't believe so. So we don't understand what is happening in that way. And the understanding of this singularity, we are not at all close to understanding what's happening there. We know that there should be a good description, but we don't have it. And why that's important is that, well, we want to know what's happening inside the black holes. Of course, we can send somebody and find out what happens to that infinity, but that would not be a fair thing. And even if we did that, even if we said, well, we send some creature, not us, the robot or something, the robot cannot come back to tell us what happened or what they saw. So because nothing can escape from black holes. So it won't be useful. So we have to figure it out without going inside the black hole, what's going on there. And that's still, we have no, not only we don't have observational data, we don't have enough theoretical understanding. And we think it's important, not just for the black holes, but it turns out that those infinities that we see, if you try to pass through those infinities to the other side, so to speak, it turns out it looks very much like the infinities we see at the beginning of our own universe. It is as if our universe is emerging from inside one of these black holes.
Along those lines, and don't lose your train of thought there, oftentimes people think of the center of the black hole as a location in space, the center. Is that the right way of thinking about it?
No, actually, it's like in a given time. It happens at a given time, and that's because the nature of space and time changes as you enter the black hole, when you cross the event horizon of the black hole.
So one of the reasons why you can't get out is because you can't stop time from ticking forward.
Exactly.
So going forward and forward in time is like going closer and closer to the singularity.
Center. And then at a certain time you hit the singularity, that's the time. It's at a given time is that you have
So the singularity could be the end of time in some sense.
It could be, but we don't believe that. And then you would say, what's the after that? And that looks very much like what could be a new universe or something. So we are interested in understanding the resolution to the issue of singularity of black holes. And I think it's very exciting. We know that black holes, as we discussed already in this program, there are a huge amount of evidence that they exist, so they are there. We cannot say we're just imagining. So what is that? What is that singularity? What are we learning from it? It's one of the most beautiful, mysterious objects, I think, in the universe. And hopefully by some experiments and maybe by some theoretical understanding, we'll have some progress in that direction.
So what's your guess? I mean, if we come back and we invite you back for the, I don't know, the 2025 World Science Festival, can you give us the answer to what happens at the center of a black hole?
Maybe invite in 2050 if I'm alive.
2050, all right. Good prediction, a safe prediction. Please join me in thanking Kumran Vafa.
Thank you.
Thank you very much. Fantastic. Good night, everybody. Thank you.